this study is exploring the safety and tolerability and the efficacy in different portions of the study for an asset called TAK-981. The name, which is previously known as TAK-981, now it's known as Subasumstat. One of my goals is eventually we will make this an exciting drug and therapeutic option for patients. So this name rolls over the tongue for people very easily. Right now, it's very difficult. So Subasumstat is a first-in-class novel mechanism, and we have found preclinically that it adds synergistic benefit when combined with Rituximab in preclinical, DLBCL or non-Hodgkin lymphoma models. So the study was aimed at, it's a phase 1/2 study aimed at combining with increasing doses of TAK-981 with a standard dose of Rituximab in the all-comer non-Hodgkin lymphoma phase 1 portion, just to establish the safety and tolerability. And then when we open the phase 2 portion, which is the expansion portion of the study, then we will hone into 2 or 3 select subsets, and we will ask for, we look for efficacy in that subset. But right now we are getting, we haven't completed the escalation yet. We are still escalating. We are generating data. What is presented at ASH is the early data that we have observed, and we were excited about presenting the data to the audience. So the study was enrolling patients that are that have already received Rituximab-containing regimens prior, and they are relapsed refractory CD20 positive non-Hodgkin lymphoma patients that enrolled in this study. First of all, when we dosed increasing doses of TAK-981, the first thing that we observed was it was very well tolerated, because so far we have enrolled close to 27 patients, out of which 21 of these patients received TAK-981 or Subasumstat, once weekly for 2 weeks in a 3-week cycle, and then 6 of those patients also received twice weekly, which is day 1, day 4, day 8, day 15 in a 21-day cycle. Among these 27 patients, that completed at least 3 cycles, when we looked at the data, we haven't observed any dose-limiting toxicity. And so, we have seen some, 5 or 6 of these patients show some low, grades adverse events, which are very highly manageable. And the AEs we have observed so far is pyrexia, chills, and diarrhea, which is very typical of an interferon therapies. The reason why I say that is the TAK-981 or Subasumstat actually activates interferon signaling, activates endogenous interferon signaling. That is one of the mechanism of action. And by doing that, it activates innate immune system. And therefore, when you combine it with antibodies that have ADCC, ADCP, there is a potential for adding to that activity. And that's what we were trying to see that in the patients. So when we saw from the safety perspective, no DLTs and the very manageable, highly manageable AEs were also interferon biology related. So we were extremely pleased, because it's sort of mechanism based events that's happening. Then when we looked at this early stage of still dose escalating, we've observed 30% of the patients showed response. So among the 6 patients that responded, 2 patients showed complete responses and 4 of them showed partial responses. And remember, this is an all-comer non-Hodgkin lymphoma study. So another CR is in mantle cell lymphoma, another CR is in DLBCL, and in the PRs, we have observed in follicular, PMBCL, and a few other DLBCL subsets. So basically, we are seeing activity across the board. So what is more encouraging and what we are so excited about is during the dose escalation, which is we are bringing a very novel mechanism of action to this patient population. We have seen a very nice therapeutic window, which is, we haven't seen any dose-limiting toxicity, but we started seeing activity. So, and that's what we are so excited about, and we are excited to bring that to the audience here. The next step is to get to a recommended phase 2 dose. So which we hope to achieve in a couple of months. So once we get to the recommended phase 2 dose, the goal is to open expansion cohorts in at least some select indications, most likely select subset of DLBCL and follicular lymphoma patients, and then expand and then generate the data so that we can eventually, if the data is positive, then eventually take it to the next step. But also, in addition to that, we are committed to this, bringing this mechanism of action to a broader set of patients. So we also have another poster in this ASH meeting, which is a trial in progress poster in which we are exploring TAK-981 in combination with anti-CD38 antibody in multiple myeloma. And in this case, we are testing 2 different antibodies. One is, an internal, Takeda developed anti-CD38 antibody called mezagitamab. And also we are trying to combine, Subasumstat with daratumumab, which is an approved agent. And this study is enrolling patients who failed, at least 3 modalities of treatment that is staple for multiple myeloma, which is the IMiD therapy, the protein homeostasis therapies, and the cell therapy. So we're trying to open, we are enrolling currently. We have opened 7+ sites in US, and we are looking forward to exploring that antibody synergy hypothesis outside of Rituximab with daratumumab in the multiple myeloma space. There is one more poster that is presented in this ASH, and it is in collaboration with MorphoSys. I hope everybody remembers MorphoSys has tafasitamab approved, accelerated approval in second-line lymphoma, the DLBCL, in combination with the lenalidomide. So Takeda scientists collaborated with MorphoSys scientists and generated preclinical data combining tafasitamab plus subasumstat, and they have shown a very nice, both in vitro and in vivo settings, very nice additive benefit and synergistic benefits when you combine these 2 agents together in lymphoma model systems. So that data is very exciting. So we are happy to present that data as well in this ASH meeting. So overall, you can imagine, right? We are very excited to bring this novel mechanism of action to the lymphoma patients. But from the Takeda company perspective, we're excited to bring this mechanism to all the cancer patients. So we do have another study, which is a phase, which is a pembro combination study in solid tumor, and we also have a single agent study that is currently in phase 2 portion, treating solid tumor and lymphoma patients as a single agent with subasumstat.